All right, good evening, Principles of Biology students. This is week 10 for Biology 211. I understand you just had a third exam, perhaps this week, and have your finals coming up, which is cumulative. So it would behoove you to review some of that content. Hopefully, this will be fresh for you. It is a complicated topic. I want to be so clear. We don't just know these things, um, or remember all of them perfectly, having taken these classes um, a year or two ago, as the case may be. I had to review my notes. I had to rewatch some videos to kind of refresh my own memory of this. So studying and restudying, learning, forgetting, and relearning is a normal process um, for these kinds of subjects, and that review process is what deepens our understanding and helps transfer that information into long-term memory, and help it stick around to make it um, so you can use it in your life. So, little segue into metacognition there, um, which was very helpful and encouraging to me when I was a principles of biology student and feeling a little discouraged about the overwhelming amount of information that I felt like I wasn't retaining. And so, with that little bit of encouragement, let's dive into gene expression and regulation. So how is a gene expressed? How is gene expression controlled in bacteria? And in general, we're looking at E. coli because it's a very easy to study organism. And specifically, we're looking at the lac operons and the tryptophan operons. And these are two of the most famous, two of the most well-studied um, metabolic pathways that we know about, and that's why we use them as this sort of intro biology um, example. So let's do a little highlighting. We've got our come on, lac operon on the left, and our tryptophan operon on the right, and we're simply going to fill in the chart. They've given us some information. The lac operon, the lactose operon, is catabolic. Let me keep my colors consistent here. Catabolic, that means it's breaking things down, right? Cats are kind of destructive. That's how I remember that. Um, and this metabolic pathway hydrolyzes or breaks down lactose, which is a disaccharide, into glucose and galactose, which are two 6-carbon sugars. And this is information that you'll have a little more context for once you take organic chemistry or biochemistry, and biochemistry, ideally. You'll be able to see how these things are very structurally very similar and related. But for now, we're breaking down lactose into simpler sugars, so that the body can use it, or the organism can use it as energy. Right? So catabolic, we're breaking it down and releasing energy. Anabolic, for tryptophan, synthesizes tryptophan from precursors. Tryptophan is really important because it's an amino acid incorporated into a lot of protein. So it is a building block. Amino acids are the building blocks for proteins. Proteins manifest into all of the characteristics of life that we can observe. So if you need a way to remember that again, anabolic steroids, bodybuilders, right? Um, Building things up. So that usually requires energy. Just to review a little bit of the content that you had earlier in the year, that is an endergonic process. It requires energy, right? Versus exergonic. Exergonic releases energy, and that would be your catabolic process. All right. So looking on the left, what regulatory genes are associated with the operon, and what functions does each serve? So you can do this maybe from memory. I certainly couldn't. So can I type this? Formatting's going to be weird if I do, so we're gonna just stick to handwriting. So for the lac operon, we've got lac1, lac-I, promoter. Promoter is a binding site that upregulates gene expression. We have CRP, and let me look really quick. CRP is a C-reactive protein. What is that? I cannot remember. It's made in the liver. One of those things that you hoped would be a really, um, really quick Google search, and it just isn't. I don't think that's the right CRP, actually. So we're going to disregard that. We'll move on. Apologies. CRP binding site operator. The functions of these genes. All right, LAC-I produces an active repressor. Active, capital R, repressor. That repressor protein binds to the operator in the absence of lactose. Binds operator if, not when, if no lactose. So if there's no lactose, the repressor binds the operator, it downregulates expression. The promoter is the site where RNA polymerase binds to the DNA, and binding of RNA polymerase is enhanced when CAMP interacts with standby. Okay. This is the important part. LAC1 or LAC-I, I'm not sure which is correct in this case, produces an active repressor protein that binds the operator if there is no lactose. CRP and the complex bind at the CRP site on the promoter region. As a result, the enzymes for lactose digestion are produced only when needed and only when glucose is not present. Let's see. So CRP and the complex. CRP and complex bind at CRP site on promoter. And these are responsible for regulating when lactose digestions occur. So as a result, the enzymes for lactose digestion are produced only when needed. So fairly simple concept. If we have a bunch of lactose around, or if we have a lot of glucose around, which is that component of lactose, we don't need to upregulate this catabolic process. We don't need to digest more lactose. So in either case, if there's a lot of lactose or if there's a lot of glucose in the system, then expression, do I have this right? Expression is downregulated because the repressor is bound to the operator. Yes, okay. Alrighty. On to, I'm going to keep this color coded because it's nice. TRPR and operator. TRPR produces an inactive repressor. TRPR produces an inactive repressor, protein that binds the operator only when complexed with excess tryptophan. Repressor protein that binds operator only with excess tryptophan present. So by contrast, in this anabolic process, we need a lot of tryptophan around. It's normally active, right? Yes, but tryptophan, the substrate, this thing that we're producing, also activates the repressor. Yes, yeah, tryptophan, the substrate, the product that we're interested in, when present, activates the repressor, which turns off tryptophan production. So RNA polymerase binds at the promoter site and transcribes the genes for making tryptophan as long as tryptophan is not in excess. All right, scrolling down a little bit. Oh, that was already there. Don't need that. Cool. Okay, we're not going to worry about that right now. So what are the structural genes? What structural genes are included in each operon, and what does each produce? So genes LAC-Z, LAC-Y, and LAC-A. LAC-Z produces codes for the B-galactosidase enzyme, which digests lactose into glucose and galactose. LAC-Y codes for a permease to allow lactose to enter the cell. Permease, permeability. LAC-A codes for transacetylase. Yes, transacetylase. Its function is still unclear. Genes TRPA, TRPB, TRPC, TRPD, and TRPE have the following products. So all these 5 genes code for the 5 enzymes required to convert precursor molecules, so even smaller subunits than amino acids, into tryptophan, one of the amino acids required for protein synthesis. All 5 convert amino acid, and in parentheses, tryptophan, precursors. All right, so is the operon inducible or repressible? Think back. We were talking about whether the repressor is active or inactive in its, I guess, ready state. The LAC operon on the left here is inducible. It is induced when lactose is present and glucose is low. Excuse me. Yes. We'll get into that in a second. The LAC operon is inducible, and the tryptophan operon is repressible. Is the repressor protein produced in active or inactive form? I was getting a little ahead of myself. For the LAC operon, the repressor protein produced in its active form. So this can get kind of confusing because we use like some double negative language. So the repressor, negative, is produced in the active positive form. Positive and negative, negative, it's repressed, okay? So it's produced in the active form. That means that it is repressed at baseline, and we have to induce to upregulate gene expression. So if the repressor is active, it's an inducible pathway. By contrast, tryptophan produces an inactive repressor. The repressor protein is produced in its inactive form. Under what circumstances would the lac operon be on versus off? Lac on, and this is when I was getting a little ahead of myself, but this is important. The lac operon would be off. Let's start with off. Lac operon would be off when there's no lactose in the cell. If no lactose. Lac operon would be on when lactose is present and there's little or no glucose in the cell. If I'm going to do a high arrow for high concentration of lactose and low concentration of glucose. So we're lacking glucose, we're lacking an energy source to make ATP, but we have this food source, we have lactose in the system. Lac operon turns on, upregulates the expression of those genes that allow for the digestion of lactose. Now for tryptophan, TRP. Off. Tryptophan would be off when excess tryptophan is readily available in the cell. If, I just realized I did really odd conventions here, that's fine. If tryptophan concentration is high, if there's excess tryptophan in the system. And likewise, it would be on at all other times. Because if we're low on tryptophan, we can't make a lot of protein. So it's going to be on because we need it for lots of things. All right, how are the lac and tryptophan operons similar in structure, function, or in both? Both, we're talking about regulation of expression. Both have regulatory genes. Regulatory genes that interact with the operator and shut down transcription of the structural genes. Transcription. And remember, as a review, the last video we did, we talked about the central dogma of modern biology. I'm just going to write it off to the side here. DNA is transcribed into RNA, gets translated into protein. And we are really talking about these operons are occurring, I'm going to put a star here, are occurring at the transcription level. right? At the DNA level. That may actually be a testable question if I remember correctly. So yeah, important to remember. What are key differences between the LAC and TRIP operons? So we already talked about whether they're inducible or repressible. The LAC operon is inducible. And TRIP is repressible. So again, that LAC operon being inducible means that the presence of lactose induces production of the enzymes needed for lactose digestion. And the TRIP operon is repressible. It's ordinarily on, producing tryptophan, which is needed for protein production in the cells, and it is turned off or repressed only when an excess of tryptophan is available in the cell. And I want to go look at my flashcards here. I had them pulled up. The tryptophan operon is an example of an anabolic pathway that is controlled by feedback inhibition, and the substrate is usually in an inducible operon. I had it backwards. In an inducible operon, the inducer is often the substrate in the pathway being regulated, and the inducer binds to the repressor, thus rendering it inactive. So we'll come back to that. Had it a little bit backwards maybe when how I was talking about it, but yes, because the substrate is the thing that yes, okay. I had my language a little bit off there, but we've been otherwise consistent. When there's tryptophan around, we repress expression of the genes that produce more tryptophan. Okay. More key differences. The LAC operon is controlled by both a regulatory protein that interacts with the operator, controlled by, I'm going to go ahead and move this over. Regulatory protein that interacts with the operator and blocks RNA polymerase action. POL, polymerase action. And a CRP site. And a CRP site. RNA polymerase does not attach effectively to the operator unless CRP complexed with cyclic AMP is attached to the CRP site. So RNA polymerase does not attach effectively to the operator if cyclic AMP, cyclic adenosine monophosphate, and CRP and CAMP bound at CRP site. Cyclic AMP is a signaling molecule. It's involved in you'll remember those signal cascades when we talked about transmembrane transmission of information. It's involved in a lot of very fast cell level communication. Once attached, it enhances the interaction of RNA polymerase with the promoter region. Cyclic AMP levels in cells tend to be low when glucose is present. So cyclic AMP low when GLU, glucose, is in high concentrations, highly available. And as a result, even if lactose is present at relatively high levels, the second control keeps production of the enzymes for digesting lactose at very, very low levels if glucose is also present. So secondary control mechanism. Cool. Yeah, those are our key differences. Tryptophan is repressible. The product is what activates the repressor. Lactose is inducible. The lac operon is inducible, and you have these two control pathways. One for the regulatory proteins that interact with the operator to bind to block the site, and the CRP complex, which responds to the presence of glucose and achieves the same goal, which is down-regulating expression when you don't need it. What advantages are gained by having genes organized into operons? So why bother with any of this? Why all of the control mechanisms? A lot of it comes down to efficiency, right? A cluster of functionally related genes that can be basically what you achieve is a cluster of functionally related genes that are controlled by a simple on-off switch. And we're going to do purple. The advantage is you have a cluster of related genes, functionally related genes. That functionally is important. They're related in function. Cluster of functionally related genes controlled by a single on-off switch. And so as they are needed, both systems are set up to simultaneously turn on or off all of the genes required in a metabolic pathway. And this is much more efficient than having each gene under independent control. That would mean an operator or some similar mechanism for each step in the metabolic process, which you simply don't need. If you don't need TRPE, you definitely don't need TRPA, or vice versa, right? Because if there's tryptophan around, you don't need any of them. You don't need any of those products, any of those intermediate products in the pathway. Cool. We talked about endergonic versus exergonic. Like all of this, too much energy could be as much a problem as too little energy, right? We're talking about homeostasis, everything being in balance. We don't want to be producing energy that we can't store or can't use immediately if we don't need it. We also similarly don't want to be building up molecules that are energetically expensive when we don't necessarily have the energy for that, right? If there's no lactose coming in, we don't want to build up stuff we don't have the materials for. Yeah. So regulation in this is all really important. Efficiency. And if we're going to take the evolutionary angle on this, those organisms on the microscopic level that were more successful at this, that are more efficient, were able to manage their resources better, were able to reproduce more effectively in a greater diversity of environments with a greater diversity of inputs and outputs. And they were more successful and persist. Cool. Question 5. Strain X of E. coli contains a mutated lac regulatory gene on its bacterial genome, and as a result, the gene produces a non-functional lac repressor protein. You add a plasmid, which is an extracircular piece of double-stranded DNA, to these cells. The plasmid contains a normal regulatory gene and a normal lac operon. Build a model or a diagram, we might not do that, of what one of these modified E. coli cells would look like. Then answer the question and use your model or diagram to explain your answer. The diagram's not super important here, but we've got our E. coli, and we have our E. coli DNA, and then we throw in our plasmid, which is our circular DNA in here. And basically, the cell division machinery of the E. coli can be used by the plasmid to express the genes contained in the plasmid. Right? So before the addition of the plasmid, would the E. coli strain X cells be able to produce the enzymes for lactose digestion? And explain. And this goes back to, is the lac operon inducible or repressible? So the answer is yes. Yes, the LAC-I gene ordinarily produces an active repressor, right? Yes, because LAC1 produces active repressor. And it's only when lactose shows up that the repressor is inactivated. So the repressor protein inhibits production of these genes for lactose digestion, and in this case, this gene is mutated so that it cannot produce the repressor protein. Yes, before the addition of the plasmid. Yeah, the gene is mutated so that it cannot produce the repressor protein, so it's always upregulating those genes that digest lactose, even when we don't need it. Right? Cool. So X cells, no repressor, which leads to cool. After the addition of the plasmid, would the plasmid's lac operon produce the enzymes for lactose digestion constitutively, that is, all the time, or only when lactose was the available sugar source? Um, we should resolve the anomaly here, right? The plasmid contains a normal regulatory gene and a normal lac operon, and as a result, the plasmid's lac operon should produce the genes for lactose digestion only when lactose was available. The original question: would the plasmid's lac operon produce the enzymes for lactose digestion all the time? No. Plasmid contains normal lac operon, meaning we would have active repressors. Enzymes produced only when lactose present. After the addition of the plasmid, would the plasmid's lac operon produce the enzymes for lactose digestion? I already asked that. Question C. After the addition of the plasmid, would the bacterial genome's lac operon produce the enzymes for lactose digestion constitutively or only when lactose was the available energy source? Oh, I see. would the E. coli lack operon produce the enzymes for lactose digestion constitutively, or only when lactose was the only available energy source? So the regulatory gene on the plasmid theoretically, could produce enough repressor protein molecules to affect both the plasmid operon and the bacterial chromosomes operon. So after the addition of the plasmid, the bacterial genomes lack operon would produce enzymes for lactose digestion only when lactose was the available energy source. So regulatory gene of plasmid should produce enough repressor protein, molecules to affect both the plasmid and genetic uh, bacterial chromosomes, or interact with both plasmid and bacterial chromosomes operon. Operons. So after the addition of the plasmid, the bacterial genomes lack operon would produce the enzymes for lactose digestion only when lactose was the available energy source. Cool. Question D. If equal amounts of lactose and glucose were present in the cell, would the lac operon in the bacterial DNA be off or on in the bacterial DNA? Would the lac operon in the introduced plasmid be off or on? So 2 questions. We talked earlier about the fact that the lac operon has 2 mechanisms for control. One is lactose present, and the second one was that CAMP complex with CAP. CRP. Which is activated when glucose is present. So if they're in equal concentration, what happens? In combination with CAMP. Cyclic AMP. CRP. CRP. CRP, catabolic activator protein, is what that stands for. CAP. It is CAP. It is an activator of transcription for the lac operon. When glucose is present, cyclic AMP levels in the cell tend to be low, and as a result, cyclic AMP does not interact with CAP, and CAP is unable to bind to the DNA, and therefore, even if lactose is present, little lac mRNA will be synthesized from either the E. coli or the DNA. So when glucose is present, I'm going to put in high concentration, cyclic AMP concentration tends to be low, and as a result, cyclic AMP does not interact with CAP, which is that catabolic activator protein. And so, catabolic activator protein is unable to bind DNA, and as a result, even though lactose is present, very little, not none, but very little, lac messenger RNA, those genes responsible for creating those enzymes for breaking down lactose, will be synthesized from either the E. coli bacteria or the plasmid. Cool. How is gene activity controlled in eukaryotes? So human genes cannot all be active at the same time, right? Kind of rehashing some of this, maybe in a different way. If they were, all of the cells in our bodies would look the same and have the same function. For specialization to occur, some genes or gene products must be active while others are turned off or inactive. And this is kind of a good point to step back and remind ourselves that every cell contains all of the genetic information required to create the entire organism. And these regulatory mechanisms are what determine the identity of an individual cell, right? So this complexity arises specifically because we have these regulatory mechanisms. All right, complete the table below. We have the gene or DNA level itself and the types of control. We have, let's stick with red. Whole chromosomes or parts of chromosomes can be inactivated by DNA packing as heterochromatin. Hetero, one word, chromatin. So it's in the word prefix hetero, many, chromatin, chromosomes. Transcription from DNA can also be controlled by enhancing or inhibiting the action of various proteins that interact with the DNA and or the RNA polymerase to make it more or less likely to produce an mRNA transcript. We have transcription. Can be controlled by enhancing or inhibiting. I could just say polymerases, but I want to make sure you guys know DNA slash RNA polymerase, which would make it more or less likely to produce an mRNA transcript. Increase or decrease of mRNA. Awesome. Then we have the mRNA product of the gene level. So here we're at, I'm just going to put this off to the side again, transcription level, and here we're at translation level, is where each of these lines are kind of occurring. So at the gene or DNA level, we then we have some control mechanisms there. After transcription, we have the mRNA product. We'll do this in a different color. At the mRNA level, controls can affect whether the pre-mRNA is processed into mRNA, how long the mRNA is active in translation, whether the mRNA is capable of attaching to ribosomes and being translated. So controls can affect mRNA processing to mRNA, how long mRNA is active, how long mRNA is active in translation, and whether mRNA is capable of attaching to the ribosomes and being translated. And ability of mRNA to bind ribosomes. And ribosomes, remember, are part of the cellular subunits responsible for combining amino acid subunits with mRNA, or excuse me, mRNA is translated into protein using the machinery that is ribosomes, which combine amino acids based on the codon of the mRNA into polypeptides, which manifest into proteins. We'll go blue. The protein, now we're after translation, we're into the protein product of the mRNA. Protein products, this is what we've been talking about a little bit in the lab. Well, the protein product may be made in an inactive form that becomes active only in specific environments. For example, digestive enzymes are usually produced in inactive form and then activated only under specific conditions of pH and acidity, or in the presence of specific activator molecules. This is kind of what we've been talking about with the lac and tryptophan operons. And if produced in active form, the protein may be inactivated by the presence of a molecule that changes its allosteric form. Allosteric just meaning shape. Basically it's shape. Or the protein product may have a very limited lifespan, and as a result, have a limited time of functioning in the cell. If we leave a protein in the cell for a very long time to do its work, has lots of substrate to do its thing, produce a lot of product. Just give it less time, right? Make it ephemeral. Protein products. Active or inactive forms. I.e., and I know I just explained all of that, but we're going to use the language that we've been learning and I guess this would be backwards. Well, no, that's fine. Inducible versus repressible pathways. But looking at individual genes. Yeah. And similar to, but unlike the lac operon and the tryptophan operons, at the protein level, any number of factors can control this. Temperature, pH, the availability of substrate, of end product. Moving right along. Ah, single-celled organisms, such as amoeba and paramecia, often live in environments that change quickly. Which of the following types of control allow organisms like amoeba to respond most quickly to frequent short-term environmental changes? And explain your reasoning. And I guess we're meant to select some here. So would control of mRNA transcription from DNA work? Maybe. B, control of enzyme concentration by controlling the rate of mRNA translation. This is interesting. So maintaining a number of different enzymes in the cell is energetically efficient only if the short-term changes occur frequently over time. Only control of enzyme concentration by controlling the rate of mRNA translation. Maintaining a number of different enzymes in the cell is energetically efficient only if short-term changes maintain many types of enzymes if changes are very frequent. Okay. C, control of activity of existing enzymes. This would respond very quickly to short-term changes in environment. So would respond very quickly to short-term changes. Sorry about the formatting on this. It's a little tight, but we do what we can. So just to make it clear, C is blue, B is red. And then control of the amount of DNA present in the cell. So in general, answers to this question will vary depending on how frequently the changes occur and how one defines short term. For example, overall, controlling the activity of existing enzymes allows the organism to respond very quickly to short-term changes in environmental conditions. Right? However, maintaining a number of different enzymes in the cell constantly is energetically efficient only if the short-term changes occur frequently over time. Um, if the changes occur infrequently and do not result in the death of the organism, but rather reduced function, then it's energetically more efficient to instead control the rate of mRNA transcription from DNA. Right? Yeah, we're gonna leave that. Depends on frequency of change and we'll say severity. Severity meaning is it an existential threat to the organism? All right, so now we're moving on to the cell cycle. And this is harkening back to meiosis and mitosis, right? Checkpoints in the normal cell cycle prevent cells from going through division if problems occur. For example, if the DNA is damaged. What forms do the checkpoints take? That is, how do they control whether or not cell division occurs? I'm going to give myself a little bit more room here because my handwriting was a disaster. We have a variety of cyclins. That are produced during interphase, that is between phases. Three of the major checkpoints in mitosis, three major checkpoints in mitosis include G1, G2, and M phase checkpoints. These appear to be controlled by cyclin-dependent kinases, CDKs, cyclin-dependent kinases. And the ratio of CDKs to their cyclins determines their activity. The activities of the different activated CDKs control the cell cycle. All right. So CDK to cyclin ratio determines activity. Activity of different activated CDKs controls cell cycle. The cell can move past a given stage of the cycle only when the appropriate checkpoint CDK has been activated. Cool. In the space below, now we're going to get a little more visual with this. Develop a handout or a diagram to explain how these checkpoints work under normal conditions. Your diagram should include a description of each checkpoint, where it acts in the cell cycle, and what each does to control cell division. I've given myself a little checklist as we go here with some reminders for myself. We're going to start with G1 phase. This is the growth phase of the cell cycle. And I'm going to draw this in terms of a clock. Okay, we're not starting at noon. We're starting at G1, which in this illustration is starting around 3 o'clock. Okay, we have our growth phase. We have our S phase. This is our DNA synthesis phase, right? DNA synthesis phase. Next, we have our G2 phase. G2 is going to take us approximately to 6 o'clock. The arrangement in space here is meant to be approximate to how long each phase takes, but that's not, it's obviously not perfect. This is a hand-drawn model. Don't quote me on that. And then M phase. This is mitosis. G2 is our another growth phase, and it's preparation for mitosis. We're getting all the machinery of mitosis ready. And then we have mitosis, which is our cell division. And at the end of mitosis, you will have, remember, your 2 daughter cells. So that is where replication happens. Now, I put this in red because it's important. It's probably the most important checkpoint to know. The G2 checkpoint, sometimes called the G2/M or G to M checkpoint, is right here. How do I want to draw this? I'm going to label it here. G2 checkpoint. Now, the G2 checkpoint is important because it checks for damaged DNA prior to mitosis, prior to replication, which is a big deal because if an error carries through into mitosis, then you're going to end up with replication of that error that will then continue on into the rest of the organism, right? So an error catching errors at that G2 checkpoint is especially important for limiting the impact of those errors in the DNA. Okay, so that's our basic cell cycle. Now let's talk again about some of those regulatory cyclins. Okay, in G1, and I'm going to use the same shape that they give here because it's useful. I'm going to make this a different color. Pen properties, give me orange. Thank you. This is your cyclin-dependent kinase. And that cyclin-dependent kinase is just going to be carried around through this cycle, okay? When we get to G2, we've still got our little cyclin-dependent kinase with its little notch in it. But we also have an accumulation of cyclin. Coming to party. In mitosis, that cyclin-dependent kinase and cyclin with its corresponding pieces come together to form their active MPF, or maturation promoting factor. Memorizing the names of these is helpful when we look at these abbreviations, because they really tell you what they're for, and you can kind of intuit where they belong in this process. Maturation promoting factor, we're trying to mature the cell, right? Shows up in mitosis because we have a complete cell that we are then trying to move along in this process. You have a mature cell, right, that is dividing. We're not replicating DNA. We're not, we're not synthesizing DNA as during the S phase. We're not building the machinery for that as in G1 phase. We're ready for a complete cell. So that's where that IPF shows up. That's how I remember it. Whether or not that is correct is its own question. Now, when we get into this next step here, it's important that this regulatory machinery, I'm actually going to get rid of that coloring so that we can see these lines. It's important that these pieces break down. It's important that it not continue working, because then we would end up with unregulated cell division or replication, or we'll talk about that in a second. Cyclin is degraded into a bunch of partial pieces, and the cyclin-dependent kinase continues on through this process. Right? Gets recycled. Hope that makes sense. We could also, as we're looking at this clock diagram, it's kind of important to think about the relative concentrations of these regulatory subunits, this cyclin concentration and CDK and MPF relative to each other, right? we can talk about MPF in terms of its activity, right? Its activity is going to be highest during mitosis, right? MPF activity is going to be highest during mitosis when cell division is occurring. Cyclin is accumulating, so increasing as we get through G1 and S phase into G2, into the second growth phase. Cyclin drops off because it's degraded at the end of mitosis, right? And I drew this arrow here to indicate CDK and cyclin. Yeah, you can see that. CDK and cyclin come together to form that MPF, that maturation promoting factor. Okay. What happens when these regulatory steps go awry? Cancer results from uncontrolled cell division. Explain how mutations in one or more of the checkpoints might lead to cancer. The basic function of each checkpoint is to determine whether or not the cell is functioning normally. Basic function of each checkpoint is to confirm normal cell function. And if it is, then it should enter and continue on through division. If yes, division proceeds. If these checkpoints break down, they could allow any cell to continue through to the cell cycle. That is to divide. So cancer is uncontrolled cell division, right? So if checkpoint the checkpoint breaks down or is absent, uncontrolled cell division occurs, which is by definition cancer. Cancer can result from a breakdown of the operation of many of these checkpoints. And without giving specific examples, I know that this is why a lot of modern cancer treatments and cancer research have a really high emphasis on individualized gene therapy, or looking at the specific genome of the cancer that you're trying to treat, because different treatments, different cancers resulting from breakdowns at different checkpoints respond better to different treatments that we have available. So this is a really interesting area of study with a very high potential for impact. So if this is something that you are interested in learning more about and having an impact on humanity, I would strongly encourage you to go down that path. And to do that, you got to do well on your exams to get to the next step. So play that game, get those points, submit your worksheets in time for extra credit. We will be compiling all of that. All of that will be done by Tuesday. So whatever the deadline is, get it in, and all of those points will be applied next week. So good luck. Thank you all for your attention. It has been an absolute privilege to go back over some of this material with you, and I look forward to seeing you all next term for 212.